Hello, and a huge welcome to everyone tuning in.
It's wonderful to be here.
Whether you're commuting, pipetting in the tissue culture hood, or, just hiding from your inbox for a bit, we are so glad to have you with us for this deep dive.
Absolutely.
Today we are doing something a little different, a special journal club edition.
Right.
Usually we take a broad topic and sort of swim around in it, but today we are laser focused. We are bridging that massive gap between high-level stem cell biology and, well, the absolute magic of how life actually begins.
It's a fascinating intersection. We're looking at the very, very first few days of human existence.
Exactly. We are talking about a major breakthrough in biology, the discovery, and this is maybe even more impressive, the creation of cells that mimic the very earliest stages of human development, specifically the 8-cell embryo.
This is a significant leap forward. And just to orient everyone, especially the postdocs and researchers in the audience who want the specifics, we are basing today's discussion on 3 landmark papers.
Let's get the credits on the board.
Right. So first we have 8C-like cells capture the human zygotic genome activation program in vitro.
Okay.
This comes from the lab of Dr. Wolfreich at the Baberham Institute, and it was led by Jasmine Taubin-Schmidstowers.
Heavy hitters right out of the gate.
Indeed. Second, a paper called Rolling Back Human Pluripotent Stem Cells to an 8-Cell Embryo-like Stage.
Rolling back, I like that.
It's very descriptive. This is from the lab of Dr. Miguel A. Esteban at the Guangzhou Institutes of Biomedicine and Health, led by Emlyn Abdul-Mazid.
Okay.
And finally, recapitulating early human development with 8C-like cells from the labs of Dr. Jichang Wang and Dr. Tuambo Sun at Sun Yat-sen University, led by Xu Yu.
Wow, so we have 3 different groups, all largely converging on the same incredible discovery.
We do, and the mission statement for this deep dive is really to clarify how these papers collectively report the discovery of 8-cell-like cells, or 8CLCs.
The 8CLCs.
And this is, it's essentially a new tool that allows us to study the black box of human origin without needing to use actual human embryos.
Which is a huge deal.
It carries significant ethical and, frankly, technical limitations. So this is a big step.
Okay, before we get into the nitty-gritty of the chemicals and the gene sequencing, I want to set the stage. Because I think most of us, even those of us who love biology, have a pretty simplified view of how an embryo starts.
It is often oversimplified, yeah.
Right. We think sperm meets egg, boom, fertilization, and then the DNA just takes over and builds a baby.
Right.
But have you ever actually wondered how an embryo knows when to start being an embryo?
That is the fundamental question.
It starts as a fertilized egg, sure, but when does it actually turn on the engine? And I guess, why does the cell even need to do this? Why isn't the DNA just on from the very beginning?
Well, to understand that, we have to look at the status quo of a fertilized egg. When a sperm and egg fuse to form a zygote, the new genome, that unique mix of mom and dad, is initially quiet.
Quiet.
We call it quiescent. It is, for all intents and purposes, silent.
So the library is open, but no one is reading the books yet.
The library is actually closed. That's a better way to think of it. The cellular machinery is running on maternal supplies. Things like RNA and proteins that the mother packed into the egg beforehand. The embryo's own DNA hasn't started doing anything yet.
So it's running on a battery pack that mom provided.
Precisely. But that battery pack runs out eventually.
Right.
And this silence has to be broken by an event we call zygotic genome activation, or ZGA.
ZGA.
This is the moment the embryo takes control of its own destiny. It's when its own genes wake up.
And this is where the species difference becomes a huge headache for scientists, isn't it?
It's a major hurdle. In mice, which are the standard model for almost everything in biology,
Right, everything, yeah.
this ZGA happens very, very quickly. It happens at the 2-cell stage.
So the mouse embryo divides once, and boom, it wakes up.
Exactly. Mice are always in a rush.
They really are.
But in humans, the major wave of activation happens later. It happens at the 8-cell stage.
The 8-cell stage. So what day is that?
That's roughly day 3 of development.
3 days.
Ah.
So for 3 whole days, the human embryo is just coasting.
It's essentially dividing without significant input from its own genome, yes. And the problem, the big problem for science is that we know a lot about mice. We have 2-cell-like cells, or 2CLCs, in the lab.
Right.
But until these papers came out, we knew very, very little about the human equivalent. Accessing human 8-cell embryos is ethically difficult and technically incredibly challenging.
It's a complete black box.
A black box.
Okay, let me try an analogy here just to make sure I've got this right.
Go for it.
Imagine the early embryo is a spaceship sitting on the launch pad.
Okay.
The systems are all off. The crew, that's the DNA, is strapped in. But they aren't touching the controls. The ship is just sort of vibrating, maybe venting some steam, running on ground power.
I like it. The ground power is the maternal supply.
Exactly. So zygotic genome activation, ZGA, is the moment the main thrusters ignite. That's the moment the ship switches to its own internal power and actually lifts off. If that ignition fails, the rocket, the embryo, it doesn't go anywhere. It just sits there and eventually fails.
That is a very, very accurate representation. In clinical terms, that failure to ignite results in developmental arrest.
Which is a common cause of early pregnancy loss.
A very common cause, yes.
And the gap in our knowledge, the big gap, was that we had stem cells in the lab that were like the crew after they'd already reached orbit. We call those naive stem cells, right? They represent the blastocyst stage, which comes later.
Correct. Naive stem cells represent the pre-implantation epiblast, so that's roughly day 5 to day 7.
So we had the crew in orbit, but we had absolutely no way to study the moment of ignition.
None.
We didn't have the cells representing that 8-cell stage launch until now.
Until now. And that brings us to the core discovery in these papers.
Okay, let's dive into section A, the discovery. This is the needle in a haystack model.
It really is.
I want to start with the findings from the Babraham Institute team, Taubensman-Stowers et al.
An excellent place to start.
Because what I love about this is that they weren't trying to engineer a Frankenstein cell from scratch initially.
No, not.
They just looked really, really closely at what they already had. They found these 8-cell-like cells just hiding in plain sight.
They did. They were analyzing cultures of normal, naive, human embryonic stem cells. And these are cells that are supposed to be, uniform, a homogeneous population.
Yeah.
But they noticed that a tiny fraction, about 1.6% of the cells, were behaving differently.
1.6%. I mean, that is a margin of error in some experiments.
It is so easily overlooked. But they used a technique called single-cell RNA sequencing, or cRNA-seq.
Which is, okay, let me try. It's like looking at the receipt for every single shopper in a grocery store.
Yes.
Instead of just looking at the total sales for the day.
That's a perfect analogy. Instead of a bulk average, you see the transcriptome, all the messages being sent from the DNA of every single individual cell.
And that's how they saw them.
And they found this small population had a distinct transcriptional signature.
Okay, let's use the bridge method here, as you call it. You said transcriptional signature.
Yes.
So imagine a crowded room. That's our cell culture.
Right.
Most people in the room are speaking modern standard English. Those are our naive stem cells. They're all saying things like, I am a blastocyst, I am pluripotent.
Correct. They're speaking the language of a day 5 embryo.
But then, this 1.6% of the people in that room, they're suddenly speaking Shakespearean English.
I love that.
They're using words and grammar from hundreds of years ago. That is the 8-cell signature. It's an older, more primitive language.
That is a brilliant way to put it, and that's exactly what they saw. These unique cells were expressing genes that should have been turned off long ago.
The Shakespearean genes.
The Shakespearean genes. Genes specific to the 8-cell embryo. The team identified some key markers. ZSCAN4, LEUTX, and crucially, a gene called TPRX1.
TPRX1. Okay, I saw this gene mentioned all over these papers. Why is it the headline act? What makes it so special?
So TPRX1 is part of a family of transcription factors called the ETCH box family. But what the Babraham Institute team identified is that TPRX1, along with a histone variant called H3Y, are highly specific protein markers for this state.
This is like a unique flag.
It's the flag in the ground. Essentially, if you see TPRX1 protein being made, you are almost certainly looking at that Shakespearean 8-cell state.
So they found these weird cells speaking Shakespearean.
Yeah.
But then they noticed something else, something even weirder. These cells weren't just using old words. They were reading from the restricted section of the library.
You are referring to the junk DNA.
The junk DNA, which we know isn't really junk. We're talking about transposable elements. These cells were expressing things that are usually kept under lock and key.
Yes. The study specifically highlighted the expression of HERVL and MLT2A1.
Okay.
These are types of retrotransposons. They're essentially ancient viral remnants and, jumping genes that our genome has accumulated over millions of years of evolution.
And in most healthy adult cells, these things are silenced, right?
Absolutely. They are buried under layers of epigenetic concrete because you do not want them jumping around, causing mutations and wreaking havoc.
It's like the cells are accessing a dusty ancient library that modern cells keep locked because the books are, frankly, dangerous.
But, and this is the absolute key, These specific elements are naturally active during zygotic genome activation in the real embryo.
Wait. So the embryo wants to read the dangerous books?
It seems so. The activation of these specific retrotransposons, like HERVL, is a hallmark of that ignition moment we talked about.
So it's not a bug, it's a feature.
It's a feature. Their activation confirms that this 1.6% of cells aren't just dying or confused. They are genuinely reverting to a primitive 8-cell state where that jump DNA is open for business.
Okay, so Taubenschmidt-Stowers and the team found them. They exist. But 1.6% is. That's hard to work with. If you want to run drug screens or big experiments, you need more than a handful of cells.
You need a way to manufacture them. You can't just hope they appear.
Which leads us to section C, making more of them. How do we turn the 1.6% into, say, 80% or 90%?
This is where the other papers come into the kitchen with their recipes.
The recipes, I love it.
And we can think of it as two different types of recipes. The first is a genetic approach.
Okay.
Taubenschmidt-Stowers et al. Found that if they forced the cells to overexpress a transcription factor called DUX4, it pushed the cells into this 8-cell-like state.
So they genetically grabbed the steering wheel and just forced a U-turn.
A very aggressive U-turn, essentially. But the other teams, specifically the lab of Dr. Miguel A. Esteban in Guangzhou, took a chemical approach.
The cocktail approach.
They wanted a cocktail that you could just add to the media, to the water, so to speak.
The magic potion. So what was in it?
They used a combination of two specific small molecule inhibitors, TSA and DZNEP.
PSA and DZNEP sounds like a bad 80s synth pop duo. What do they do?
So TSA is a histone deacetylase inhibitor, and DZNEP is an EZH2 inhibitor.
Okay, let's unpack that. That's a mouthful.
Yeah.
Analogy time.
Let's do it. Think of the stem cell state, our modern English speakers, as being inside a walled garden.
Okay.
The cells are happy there. The walls are made of epigenetic modifications that prevent the cells from, sliding back down the hill to the 8-cell valley.
Okay, so the walls keep them modern.
Exactly. And one of those walls is histone deacetylation. It's a process that tightens how the DNA is packaged, which effectively closes the book on those old 8-cell genes.
Right.
TSA stops that tightening. It inhibits the deacetylases so the DNA relaxes.
So TSA is like a chemical that dissolves the mortar in the wall.
Precisely. And DZNEP stops a different process, a form of methylation that also keeps those genes off. So together, the chemical cocktail dissolves the cement, holding the walls up.
And without the walls?
The cells naturally roll back down the hill to the 8-cell valley.
That is incredibly cool. You just remove the barriers and the cells, they want to go back to the beginning.
It appears to be a more fundamental state, yes. The Guangzhou team achieved very rapid conversion using this method.
And they found something else, right? A mechanistic insight.
They did, and this is really important. They identified a factor called DPPA3.
DPPA3?
Yes. DPPA3 is a DNA methylation regulator, and they found that when you use this cocktail, DPPA3 goes to work and helps actively remove the DNA methylation, those off switches, allowing those 8-cell genes, like our friend TPRX1, to turn on.
So DPPA3 is the demolition crew that comes in after the walls are weakened.
A very fitting description. But there was a catch.
Always a catch.
The third team from Sun Yat-sen University, led by Zhu Yu, they noticed a problem with these chemical methods.
And what was that problem?
The cells tended to die.
Yes. That's a significant problem.
It is. Reverting to such a primitive and, frankly, chaotic state is stressful for the cell. Or maybe the culture conditions weren't quite right. But the cells were undergoing apoptosis and necroptosis.
Programmed cell death.
Two different forms of it, yes.
So they were rolling back down the hill and then just falling off a cliff at the bottom.
Correct. So the Yu et al. Team added a crucial garnish to the cocktail. They added anti-death drugs, specifically inhibitors of necroptosis and apoptosis.
They put a safety net at the bottom of the hill.
Exactly. By blocking the cell death pathways, they could maintain these 8-cell-like cells for much longer, which allowed for much more detailed study.
So now we have the cells. We found them. We know how to cook them up in large batches. We can even keep them alive. But now comes the big question, the ultimate test. Are they real? Or are they just stem cells playing dress-up?
This is the crucial test, section D, the test of totipotency versus pluripotency.
And we really need to define those terms clearly because they sound so similar, but the difference is absolutely everything.
It is a critical distinction. Pluripotent cells, like the naive stem cells we started with, can make any cell in the body.
Heart, brain, liver, skin.
All of it. They can make the baby,
No.
but they cannot make the placenta. They have lost that ability. It's a key lineage restriction.
Okay, and totipotent.
Totipotent means the cell can do it all. Toti, from total. It can make the body plus the entire support system, the placenta, the yolk sac, all the extra embryonic tissues.
Got it. Baby plus support system.
And the true 8-cell embryo is totipotent.
So if these 8-cell-like cells are the real deal, they should be able to make placenta cells.
That's the test. And this is where the bridge was crossed, so to speak. Did they pass it?
Don't keep us in suspense.
They did. Both the Mazid et al. Paper and the Yu et al. Paper showed that these cells could differentiate into trophoblasts.
Trophoblasts, which are
the precursor cells of the placenta. Pluripotent stem cells generally struggle to do this efficiently, if at all, but these 8-CLCs did it.
Wow. And didn't the Yu et al. Team take it a step further? I remember reading about blastoids.
They did. This was a fascinating experiment. They showed that if you just put these 8-cell-like cells together in the right conditions, they self-organize.
On their own.
They spontaneously form structures that resemble the early blastocyst. They have the inner cell mass, which would become the fetus, and the outer layer of trophoblasts, which becomes the placenta.
That is mind-blowing. They self-assembled into something that looks like a day 5 embryo.
It demonstrates that the program is functional. It's not just a list of genes being expressed. The cells understand the instructions and can build the architecture of early life.
And just to tie a bow on the verification part, how did they make sure they were using the right cells for these big tests?
They went back to our trusty marker.
TPRX1.
Exactly. They used the presence of TPRX1 to sort and verify the cells before running these demanding functional tests. If it didn't have TPRX1, it wasn't considered a true 8-cell-like cell.
Okay, this sounds almost too perfect. We have 8-cell embryos in a dish. We can make them. They act like the real thing. They can build blastoids. But science is never that clean, is it?
Rarely, if ever.
This brings us to Section 4, the critical review. We need to look at the limitations, the discrepancies, the debates. What's the catch?
Well, the first big limitation is what I'll call the origin story.
Meaning where they came from.
Precisely. A real 8-cell embryo, a blastomere from that stage, comes from a zygote. A massive, singular cell that's packed with maternal nutrients, proteins, and RNA. It has a huge volume.
Right. The egg is the biggest cell in the human body by far.
Exactly. But these 8-cell-like cells, they come from stem cells, they're tiny, they have divided countless times in the dish, they have no maternal factors left.
So physically
and metabolically, they are quite different from a real blastomere.
So they're reading the same script, the same Shakespearean play, but one is a massive, well-funded opera house production.
Yeah.
And the other is a minimalist black box theater performance.
That is a fair analogy. The transcriptomic signature, the script, is the same, but the physical context is different. And we don't yet know if that size difference or metabolic difference matters for certain downstream developmental processes.
Okay, that's a big one. Then there's the issue of stability. You mentioned earlier that the Babraham team found these cells were transient.
Yes. Taubenschmidt-Stowers noted that in their normal culture, the cells cycle in and out of this state. It's unstable. It's a fleeting moment.
And you ever had to use those anti-death drugs to keep them alive.
Exactly. Which raises a really important question for the field. Is this a stable cell type that we can keep in a jar, or is it a state that cells must pass through?
Are we freezing a frame in a movie, or are we building a statue?
That's a deep and very accurate question. If biological time is constantly moving forward, can you ever truly park a cell at day 3 and have it stay there? The jury is still out.
Right. And there was another issue, a discrepancy between the papers.
The recipes.
The different cocktails. Mazid used TSA and DZNEP. You had a slightly different mix. Taubenschmidt used genetic overexpression of DUX4.
It's like 3 different chefs claiming they have the perfect recipe for beef Wellington.
And they all taste slightly different.
And they probably are all slightly different. This debate is healthy, you know? It suggests there are multiple roads to Rome.
Multiple ways to unlock the genome.
Exactly. But it also means we haven't standardized the perfect 8-CLC yet. We don't know which method produces cells that are the most authentic.
Now, I want to circle back to something you dropped earlier, which I think is a critical detail for our listeners who might be asking, why not just study mice this whole time?
The TPRX1 issue.
You mentioned TPRX1 is the gold standard marker for these human cells.
Yes, and here is the critical detail from the Mazid paper that really drives this point home. TPRX1 is absent in mice.
Mice. They just don't have it.
It was lost during evolution in the rodent lineage. It's gone.
Wow.
This highlights exactly why mouse models failed us in understanding the specific stage of human development. The ignition key for the human embryo is different from the key for the mouse.
So we physically cannot study TPRX1 function in a mouse.
Because the gene isn't there. It completely justifies this entire line of research. We can't just extrapolate from mice anymore, not for this.
Yeah.
We've been trying to understand human origins using a manual written for a different machine.
And these HCLCs finally give us the manual for the human machine.
That's it.
Okay, so bringing it all together for the conclusion. We have identified what I'm calling a ghost in the machine. A small population of cells, these HCLCs, that somehow remember their origins.
It is a form of cellular memory, yes. A transcriptional memory.
And by using markers like TPRX1, ZSCAN4, and LEUTX, scientists can now finally isolate, create, and study the precise moment human life activates its own DNA.
That is the summary. But the implications, the so what, are vast.
Let's talk applications. Why should the listener, at home or in the lab, care?
First and foremost, infertility.
Right. The failure to launch you mentioned.
Many pregnancies fail exactly at this ZGA stage day 3. The embryo fails to ignite its genome. Until now, we couldn't study why because we couldn't just experiment on patient embryos.
Of course not.
But now we have a model. We can break genes in these HCLCs using CRISPR and see which ones cause the ignition to fail. We can test drugs to see if they help.
That could be absolutely huge for IVF success rates.
Potentially transformative. Second is regenerative medicine. Understanding ttipancy, the ability to make everything that's the holy grail of the field. If we can truly control this state, we might be able to generate tissues for repair in ways we currently can't even imagine.
And the third one, the one that's a bit more futuristic.
Synthetic embryos. The ability to make blastoids from HCLCs. This raises serious ethical questions that society needs to discuss, of course.
Absolutely.
But it also provides an incredible platform to screen drugs for safety during early pregnancy without ever putting a real embryo or a person at risk.
It's a brave new world.
It really is.
I want to leave our listeners with a final thought to chew on.
Okay.
We talked about rolling the cells back. We talked about dissolving the walls so they slide back down the hill.
We did.
If we can chemically roll back a cell to the very beginning of its life, before it even decided to be a body or a placenta, does that mean biological time is reversible? We aren't just pausing the clock here. These researchers essentially turned it backwards. If a cell can forget it's 2026 and think it's day 3 of its existence, what even is age?
That is a profound question. It suggests that age, in a cellular sense, might just be software. It's an epigenetic program. And if you have the right password or the right chemical cocktail, you can reboot the system.
Reboot the system.
But we have to remember, knowledge is most valuable when we understand the how and the why, not just the result. We have the cells now. The next decade will be about understanding the wisdom they hold.
Rebooting the system. I love that. Well, that is all the time we have for this Journal Club Deep Dive. We hope you learned something new about your own origins today.
Thank you for listening.
See you next time.